Inaugural lecturer, very distinguished ladies and gentlemen. Charles Fowler once made an honest observation that the best teachers of humanity are the lives of great men. And today, we are gathered to honor greatness and to tap from the fountain of knowledge that flows without end, that fills the hunger of ignorance, and that satisfies the human soul with its endless stream. Members of the university community, very distinguished ladies and gentlemen, an applause for our 126th inaugural lecturer as he stands for his citation. It is actually on an appeal from the inaugural lecturer that I'm going to have an extended background of the circumstances of his birth and childhood. I would have loved to just round it up in a matter of sentences. Born on the 25th day of October 1962 to the Asikong royal family, his birth came as relief to his mother, who had hitherto battled childbearing difficulties. Like the fresh breath of the Nigerian independence, 2 years before his birth, the little Ernest-Bassey Etta Asikong had come to stay, and to stay for good. His mother had gone through the agony of bearing a number of her infant children before him. With this, surely, he had begun his resolve to be alive and to strive and to attain absolute greatness and to make an impact right from infancy. And it is an unassailable truth to say that before us is a man of unbreakable resolve. However, his survival through the pensive days of infancy cannot be distanced from the belief strongly expressed by his parents that he had been predestined to stay, to wipe the tears of past misfortunes. His father's repeated narration that an angel had appeared to him in the image of a long-demised relative with words of assurance on the little boy testifies to this. Today, that little boy who many had feared would face the fate of his earlier siblings stands before us, clothed in the ethereal colors of academic success, and graces the stage as our very distinguished 126th inaugural lecturer of the University of Calabar to talk about the profitability of microbes to mankind. Professor Ernest-Bassey Etta Asikong's father, late Prince Etta Asikong-Bassey, whose father ruled the Tigidi kingdom for 45 years, was a loving father and husband whose marriage to late Mrs. Adiaha Etta Asikong, both of the Tigidi kingdom, remain unshaken by the trying times. The professor is the first child of his mother in the 11 children of the polygamous family. Professor Ernest-Bassey Etta Asikong started his primary education from government primary school in Tikiti and proceeded to complete same in St. Ben's Primary School, Akbara Ikureku, in Abi local government area. From the very beginning, he proved himself to be a boy brimming with brilliance and desire for more, and of course rounded off his elementary education in 1975 with distinction. He advanced to Agbo Comprehensive Secondary School, Egbori, Akomsko as it is fondly called. And Akomsko alumni here, thank you. Remarkably, he continued on the path of excellence already established, passed his WAEC in 1918 flying colors. A year later, of course, he arrived at the University of Calabar as a freshman, where he enrolled for the then one-year remedial program known today as pre-degree. Armed with unwavering determination for success, he burned the midnight oil, which earned him not just excellent grades, but admission to a degree program in the 1982 academic session. Hence, he backed his master's, he backed his BSc degree in 1986, crowned as the best graduating student of his class, and with the only second-class upper division in biology with focus on microbiology. He obtained his master's degree in 1995 with scholarship from the federal government of Nigeria, and with a stream of knowledge ever blistering, he enrolled for his PhD with the University of Calabar State and successfully earned a degree in 2011 in environmental microbiology and biotechnology. His educational trajectory clearly demonstrates that he is a thoroughbred Malabite, having obtained all his degrees from the University of Calabar. Professor Ernest-Bassey Asikong, our lecturer, has also had an interesting work life. He started his career as a radio officer with the defunct Nigeria Rubber Board in 1981 and worked for a few months before quitting to afford him the time needed for his university education. During his NYSC, he worked in the General Hospital who carried the former Gongola State, now Taraba State, where he was engaged in laboratory analysis from 1986 to 1987, of course, but he was there. Shortly after his NYSC, he had a short stint with the Public Department of the Ministry of Health under the National Public Work Scheme for unemployed graduates. Later got engaged by the Post-Primary Schools Management Board in 1988 as a biology master, where, of course, he mentored, sponsored, and took students to different levels, different competitions, and won laureates, which earned him accelerated promotion that made him 5 years even ahead of his peers. Due to his quest for laboratory career, he sought for deployment on secondment to the UNICEF Assisted Water and Sanitation Project, now Rwatsa Laboratory as a principal microbiologist, water quality and environmental analyst. His passion to dispense knowledge gathered led him to take up a fresh appointment as a graduate assistant in 1993. From this period, he continued on a steady climb along the rungs and along the ranks until he attained the much revered position of Professor of Environmental Microbiology and Biotechnology in 2016. Professor Ernest-Bassey Asikong is married to his heartthrob, Evangelist Mrs. Margaret Ernest Asikong, who is the Director of Information Technical Services, Ministry of Information, and is now secondly on secondment, as was introduced to us here, as Editor-in-Chief, Murphy News Magazine in the Ministry of Finance. That marriage is blessed with wonderful and beautiful children, Emanuela, Favor, and Divine, all of whom are students of the University of Calabar. Over the time, Professor Asikong, our lecturer, has proven to be a loving, dedicated, and responsible husband and father whose commitment to his family is unparalleled. He's a devout Christian, and as a humble man that he exemplifies, he has demonstrably instilled in his children and family at large the inestimable values of love, honesty, humility, mutual respect, and a God-fearing disposition. Professor Asikong has also held a number of positions in the university. He has been examinations officer, he's been academic advisor, he's been head of department, he's been the chairman of Nigerian Environmental Society Cross River State Chapter, director University of Calabar Alumni, and director currently Science, Engineering and Technology Projects TEPI, which he holds for this moment. Other responsibilities, of course, that have been held by Professor Asikong, a lecturer, and recognitions and laurels also that have been won by him are actually very interesting. And of course, tell us much about his scholarship, his depth, and tell us also much about his character as a scholar. Professor Asikong has supervised over 300 students, including undergraduate projects, postgraduate diplomas, MSc, PhD theses, and dissertations. He has published widely in local and international scholarly and scientific journals with a remarkably high visibility, citation, and H-index. He has served as external examiner for undergraduate and postgraduate projects and thesis defense, external assessor to professorial cadre for a number of universities. He has also authored seminal texts, yes, including Introduction to Environmental Biology, Microbiology, Pollution and Waste Management, Environmental Microbiology, Impact Assessment, Health Safety and Environment, Microbiology and Public Health and Introductory Perspective, and so on and so forth. He has also been, yes, on the NUC accreditation to a lot of institutions. He has won a lot of awards. Let me just summarize them. Both at the NYSC level, he had started, of course, winning awards. Award of Excellence for good relationship in the department, in the faculty. He has also got an award as Environmental Stewardship Award, actually. And he has also been an environmental consultant, especially on environmental impact assessment and bioremediation. These are some of the areas that he has won awards in. His community service has seen him occupy or play a role as member University of Calabar ceremonials, the prizes and awards committee, the convocation committee, of course strategic planning committee of the university, and baseline assessment committee, and so on and so forth. Our time would not allow me, just as it's been said, to say all these things that he has been. So let me just say that he has also had a lot of demonstrations and experimentation presented during seminars, during workshops, and during research fairs. They are all in the lecture thesis that. So he has also been a consultant, as I said, to very many. Professor Asikong, as I said, is a devout Christian, an elder of the Apostolic Church, and is very committed to the service of God in the Unical Chapel of Redemption, an interdenominational church in the University of Calabar, where he has served as Assistant Secretary of the Church Council for 2 tenures, and currently is an adult Sunday school teacher and a member of the Men's Fellowship. So, distinguished ladies and gentlemen, we are gathered here today to celebrate the zenith of academic excellence. We are here to grace the day with delightful smiles and hearts, as I can see, beaming with joy. So before us is a profoundly consummate scholar, an accomplished academic, and a trailblazer, an unalloyed administrator, an inimitable teacher, an extraordinary scientist, a loving father and husband, a professor of environmental microbiology and biotechnology, a consultant who has lived by the calling of his profession, by the calling of his career as a scholar, and by the motto of the University of Calabar, knowledge for service. Service to society, service to country, service for mankind. And here is this professor as the 126th inaugural lecturer of the University of Calabar. Thank you, and thank you very much.
This is achieved during biochemical settling, decomposition, and composition. To all of us seated here at the end of your life, you will be handed over to microorganisms, and they will return you back to where you belong, whether we like it or not. In the course of my lecture, I'm going to look at why meat, as contained in my topic, why it is fine. We're going to look at, why microorganisms are tend to be unprofitable. Why they tend to be unprofitable, we consider our unravel their relevance, various areas, especially to humanity, to the environment, and the renewable energy. We'll look at my research focus, my contributions to knowledge, and we summarize. Why meat? Why are microorganisms seen to be meat? Oh. It is with a deep sense of humility and profound gratitude to stand before this great audience of the University of Calabar and very distinguished invited guests to present the 156th inaugural lecture of this great University of Calabar. On this auspicious day of the 17th day of July 2024, Vice Chancellor inaugural lecture is an opportunity for a newly appointed professor to contribute his knowledge to the university. There is always why he is a professor and also inform the town and the town on this career trajectory. Accord the lecturer the opportunity to elucidate what he has been doing, also to give update on his research directions, and highlight his contribution to knowledge of which lecturer has earned his professorship, and then explain his future research focus. Having been appointed and promoted professor since 1st October 2016, I have therefore been owing this debt for 7 years, 7 months, and 17 days, counting on this day. And 7 is completion. It is therefore on my honor and privilege, therefore my honor and privilege to present the 100th inaugural lecture on the topic, demystifying the myth of unprofitability of microorganisms, unraveling and unveiling their relevance to humanity, to the environment, and renewable energy solution. Vice Chancellor, the topic was carefully chosen after considering how microorganisms have been so neglected by humanity and giving the common place after contributing so much to our survival and sustenance. It had been so mythified. When I was growing up, each time I saw an inaugural lecture title, I'll be confused and bite my tongue to put down the title because they were always written in coded languages, which could be decoded only by the inaugural lecturer. And I told myself, when I grow up and I want to present an inaugural lecture, I will make it so simplified so that even my community people will understand the topic. That's why the topic is so simple, and I know you have understood it even as we came in here. Thank you very much. Why me? Meat is simply a widely held but false belief. All right, here. A misconception, a fallacy, a mistaken belief, and also a false notion. It is a symbolic narrative usually of unknown origin. There is a proverbial saying that when dry bones are mentioned, the old men become uneasy. keeping them alive. The same goes for microorganisms. Whenever microorganisms are mentioned, everyone is scared because of their association with sicknesses, infections, and ailments. Most especially during epidemic or pandemic. Why do we need to demystify a bit? To demystify simply means to remove the mystery or make it rational or comprehensible. It is to clarify or clear a doubt. It is the art of making something easier to understand and less complicated by explaining it in a clear and simple way. It is on the above premise that I stand before this audience as the 126th inaugural lecturer to demystify the above me. I'm here to disparage, to puncture, to deflate, to expose, to clear our doubt about microorganisms so that no one mystifies or confuses it again that microorganisms are known for nothing good except for causing diseases. Microorganisms, what are they? If I ask me, they are simply organisms that are so tiny, so small, so myopic that we cannot see them with our own normal or natural eyes, except with the help of a microscope, which is an instrument to magnify objects. Large size that it see. As we are seated here, they are with us, even more in number than us. As you laugh, as you talk, as you smile, as you sing, as you shake hands, as you touch one another, you are releasing billions of them into the atmosphere. So beware before they come on you. Vice Chancellor Omar, let me speak to my people. Microbiology, do we kele I can give you a piece. In the way kele I can give you a piece. As I seated there, they are with you. God, I have there. Am I communicating? In every language, in my name, I'm not prophetic, but whatsoever you have there, you can interpret it. When hearing for ADD to make any good. Why am I considering tagged unprofitable? Why are they tagged unprofitable? Pastor Norman, microorganisms are so criticized as being unprofitable because of their implications as etiology of some infections and diseases. Yes, various types of microorganisms are valuable, including viruses, bacteria, fungi, algae, and protozoa. They are also implicated in causing several diseases. Samples of viral diseases or infection include measles, hepatitis, polio, Ebola, Lassa fever, HIV, COVID-19, and others. Bacterial infections include cholera and other sicknesses like typhus, diarrhea or leprosy, bacterial endocarditis, tuberculosis, gonorrhea, and several others. Fungal diseases, few of them include ringworm, candidiasis, aspergillosis, plasmomycosis. And then parasitic disease might include malaria, amoebiasis, schistosomiasis, and a few others. Time will permit me to enumerate them. Here is simply an overview of viral infection of humans. You can see almost every part of the body, is affected. Last lecture, we were told about matters of the heart. I want to tell you that in matters of the heart, microorganisms also matter. You can see cardiovascular disease caused by positive B virus and endocarditis, bacterial endocarditis, which is caused by some species that the probes. It might be. What are they? How can we acquire microbial infection? First, by contact with other people, through coughing, who are infected, through coughing, sneezing, And you can also contact microorganisms, through contact with objects. For instance, how many times have you washed the handle of your door, or the handle of your office, or anywhere, or your car handle? You open with your hand and close it on daily basis. Even in hospital wards or hospital room, patients come, open the door, and enter the consulting room. Other nurses come also open the door with their hands. Even doctors, but they are not get to wash. So, this is a very major area. Even your phones. How many times have you washed your phone? Have you ever? But you touch them with dirty hands. Sometimes with the hands, you rub your eyes, you pick your nails, you rub your ears, and you can easily contact this infection. Also through sex, and most importantly, through contaminated food and drinking contaminated water. Please permit me to look at the sustainable goal as it relates to health. Well, the SDG 2030 has 17 goals, but specifically goal number 3 focuses on health, which provides ensuring healthy lives and promoting well-being for all at all ages by 2030. Ensuring healthy lives and promoting well-being of all at all ages, requires sustainable development. It's essential to sustainable development. While other nations are striving to achieve the 2030 development goal on health, Nigeria is being plagued with cholera. And as of June 2024, it is reported that 1,579 suspected cases, have been reported. These are those who come to the hospital, what of others. So the case might be more than this, and we have 56 deaths, according to CDC 2024. How can we prevent malaria or cholera? First of all, you need to boil your water well and also purify or treat them for drinking. Effective hand washing, very, very essential. Cooking your food, very essential. Washing your vegetables, before taking it, also very essential. And also avoiding, as of now, because of the epidemic of cholera, please, you might as well avoid eating food that you cannot feel. Avoid the problem of cholera. Keeping your environment clean also very, very important. Government and other authorities should ensure they establish robust health surveillance system. They respond to outbreak easily, and then intensify public awareness campaign, which are benefiting freely. I'm briefing you on how to prevent cholera. Don't say I did not tell you. As an advocate of water, sanitation, and hygiene, permit me to share idea on how we can easily prevent cholera. Hand washing is a simple process, effective and inexpensive way to prevent diarrhea and respiratory infection. Hand washing is common, but it came to the front burner since 2008, when the United Nations General Assembly declared the 15th of October as the Global Hand Washing Day. But must we wait till the 15th of October before the whole campaign on hand washing? No, it's something we need to practice daily if we want to live and survive. What is effective hand washing? Simply, effective hand washing is the act of cleaning our hands with clean and safe water. Clean and safe water with running water and soap, preferably liquid soap. I know you wash your hand before you came here, but how effective was it? Avoid washing hands with wash hand basin. Each time you go to a restaurant and they present wash hand basin, please run away. Those ones that present tap water for you to wash your hand, please, those are the ones you should sit and eat. The hand washing basin, many people have used it, they have not cleaned it, they present it to you. As many as 100 persons who come to use the same hand washing. Some sick, some healthy, and as you eat there, you take diseases home. So hand washing is very necessary to be healthy. So the emphasis on washing cannot be overemphasized. These are some of the processes of hand washing. Without bugging you, remove your rings, the rings from your hand, whether they are wedding, no matter how important the rings are, you need to remove them because microorganisms are inside those. Before you know it, you contact diseases from those rings. Your wristwatches must be removed, and then wet your hands with running water. Take some little drop of soap, rub your hands to provide lather. The lather is very important because it acts as a surfactant, which helps to kill the microorganisms. And then the water now runs, the microorganism down. So as you wash the basin, you wash back the microorganism, take it back to your hand, and then you end up doing nothing at the end of the day. So time is going to be, you can take this from my manual. What is the way forward? Must we wait until there's epidemic or pandemic before we practice hand washing? During COVID-19, all of us had buckets with tap. Am I lying? You had it before your door. And because there's no COVID-19, you have thrown it away. Please return it back because it will help you most sincerely. So, we must bring back the practice of hand washing. We want to encourage it. Let government establish hand washing stations in schools, in marketplaces, and public places so that people can be aware of this. Pastor Solomon, permit me to state the obvious. That it will not be an overstatement. The obvious that humanity, mankind, and the environment would not have been sustained after creation without the activities of microorganisms. These are places that we move to, and in the course of allowing the land to follow, it encourages biodiversity and natural vegetation returns and support various plants and animals. So microorganisms will circle the different over and over again. Particularly in nitrogen cycle, organisms such as Rhizobium, Bacillobacter, Pseudomonas, Nitrobacter, Pseudomonas, and Clostridium are the organisms that help to circle this nitrogen, especially during nitrogen fixation, nitrification, and also denitrification. These microorganisms are very, very important to help to recondition the soil. Nitrogen is very essential to plants. Microbes are also very essential as biopesticides. Pacillobacter specifically is used to produce insecticides. Instead of using chemical insecticides, Pacillobacter as a bioinsecticide is most environment friendly. So, and have also been used to control malaria in several nations. As probiotics, microorganisms or probiotics are simply living organisms that we can take in because of their health importance, especially from yogurt. When you culture yogurt, with some microorganisms such as Lactobacillus and Streptococcus thermophilus, they are in there. As you consume the yogurt, you take in this microorganism because of their beneficial effect. So I want to strongly encourage you to consume yogurt and cheese because they will help you very strongly, just like that young boy is enjoying himself. Medicine, pharmacology, and pharmaceutics, a wide variety of microorganisms have been employed in the production of drugs. Microbial world is the system that is divided against while others are causing diseases, others are producing substances that can confront and kill the others. Particularly penicillin is the first antibiotic that was produced and was learning from a penicillium or a fungus known as penicillin notatum. Several other antibiotics have been produced. This death can get others from manual. In cassava processing, most importantly, cassava processing is an area that microorganisms play a very essential role, especially my community that I take in that book. It contains cyanic acid, which is very poisonous. And so you need to process the cassava leaf very well by washing it very well before taking so that you don't have the poison. But most importantly, the pest. Microorganisms play a very essential role in removing the cyanic acid from the pest. Microorganisms such as Bacillus, Deuterium, Candidum, Corynebacterium, Manihodium, Rosarum, Aspergillus, Penicillium, Saccharomyces cerevisiae, play a very important role in breaking down the cyanic acid so that your cassava becomes less poisonous. Avoid any cassava or gari where the cassava is harvested today, processed today, and fried into gari today, or also turned into food today. When you allow it to ferment for 3 to 4 days, it allows microorganisms to break down the salicylic acid, and eventually the chemical, the salicylic acid, is removed from it. So be careful and avoid eating unproperly processed garlic. A single cell protein which can mitigate the food crisis, there are several microorganisms that can be utilized, including a few of them on the board. And it will be also microorganisms that are the real superpowers of the world. You agree with me that never a time has any world power with the most powerful military armament of weapons been able to terrorize the world like a small virus called coronavirus. The two world superpower went into hiding, or did you not hide during that time? Yeah, I went and did myself. Even their presidents were infected but narrowly escaped death. Remember the British president, even American president, had it. You know, they were turned, many of us were turned into masquerade, even medical doctors, because of being afraid of very small organisms. The entire world was locked down, the streets empty, schools closed, markets shut down, offices locked up, industries shut down, even churches were not spared. All of us intensified our prayers in order to be spared. Some of our students seated here, we would have graduated 2 or 3 years ago, but COVID-19 is still keeping you in school. Oh. One may therefore conclude that microorganisms are the true superpowers of the world, but therefore not be underrated. This is the statistics of COVID-19 prevalence. This is the only time Nigeria was better than other countries. You can see while Nigeria is still in 100, United States, India, France, and Germany are ranked very high in occurrence or maybe infection of COVID-19. Genetic engineering is also an important area where microorganisms play an important role. We have heard of genetically modified organisms and many foods that have, high quality in terms of protein content and other nutrient content. And in genetics, microorganisms play a very important role. Also in the environment, microorganisms play a very important role, especially in waste management. You can imagine, since we were born, that the waste generated by our forefathers and even us were allowed to litter the streets. There would have been no space for us to build houses or establish schools or industries. But as we gather this waste and stop them somewhere, microorganisms break them down and return them back in the soil. Irrespective of the status, like I told you, microorganisms return all the organic components back to the earth through waste management. These are simple processes of waste management which time will fail me to elucidate more. It's in the manual. This is a typical waste dump in Nigeria. If you look closely, you know the environment. While this is potential wealth lying down there, it is also risky. In the next 50 to 60 years, there will be explosion in this waste because methanogenetic microorganisms are still acting on the organic component underneath. Methane is being formed, and when it exceeds capacity, it will explode. And so while it is well, it is also dangerous. In global warming, microorganisms have played a very significant role. How? Because they have high potential of biophotosynthesis. So they remove carbon dioxide, which is a major component in biophotosynthesis from the atmosphere. Carbon dioxide is a greenhouse gas in the atmosphere. So microorganisms, especially cyanobacteria and also microalgae, help to remove carbon dioxide from the atmosphere, thereby reducing the carbon dioxide component and then helping to control global warming. It helps to sequester carbon dioxide. Japanese scientists are working on a microalgae, which has the capacity to take carbon dioxide from the atmosphere 50 times faster than green plants. And so it is by so doing that will help to control global warming, and that is where results will be achieved in controlling global warming. Microorganisms are very important in my regulation of petroleum industry, especially in hospitalities. They also act as environmental monitor, especially during environmental impact assessment. They also play an important role in plastic control. Plastics are non-biodegradable, but microorganisms have the capacity to break down the plastic, the calcium bonds, so to speak. Pastor Soma, unraveling the relevance of microorganisms in renewable energy, it is a way of trying to mitigate the selling of the sustainable development goal 2030, which provides that we ensure access to affordable, reliable, sustainable, and modern energy for all, clean energy for all by the year 2030. It is in attempt to get into this goal setting and finding solutions to the energy crisis in Nigeria that has informed my interest in biogas, which is the source of renewable energy. Why did I focus on this? The reason being that the technology gap and the energy crisis in Nigeria can never be mitigated except we make the effort to develop our indigenous technology with respect to availability of raw materials and also resources. There's no white man that can transfer his technology to you, because that is where his power, political power lies, that's where his economy lies, that is where his security lies. You can imagine a white man that has developed a technology that can, or even microorganisms that can even destroy the world within seconds. They can never sell the technology to you. So we need to develop our own indigenous technology. That was what informed my interest in biogas. Well, what is renewable energy? Renewable energy that is not replenishable. It is sustainably available for us. So long as you tap into it, you can always have your energy. Some examples of renewable energy as listed above, but most importantly, biomass is a major source of renewable energy, that is biogas calculated from biomass, which is simply organic plants and animal component. What is biogas? Simply then is gas generated by fermentation of biological components by microorganisms under oxygen-free environment, especially under anaerobic, what is called anaerobic environment. As we are here, we are in aerobic environment. There's oxygen available, but there are places where you don't have oxygen available at all, and that is where microorganisms that generate biogas, that is where they thrive more. This is simply some of the components of methane, 57%, the biogas generally has 57% methane, then 30 to 40% of carbon dioxide and other gases in various elements, as can be seen. This is simply the molecular geometry of methane, one carbon and common goes hydrogen. Time will fail me to elucidate more. This is the flow chart of biogas production. Simply collect their waste from any source, any organic component can be used, process it, inoculate it into an anaerobic digester, and then within the process of time, as we inoculate microorganisms, so to speak, including metallurgists, they break them down, to biogas and slurry, which is two of them are very essential. These are simply the processes biogas production follow. There are 4 basic processes, including hydrolysis, acidogenesis, acetogenesis, and then methanogenesis. It's like a relay. Hydrolysis hands over, and gradually methane is produced by methanogenic microorganisms. Please, the details are contained in my manual. Major substrates used in biogas production during my primary research include water lettuce and water lettuce or water hyacinth. When I was growing up, each time I went to my fish pond, the fish pond in my community, I usually, you have water hyacinth covered the whole stream. When I used to go to fishing with my elder brother, as the layman, there will not be any space to put the line. So when I was researching, I said, how can I find a solution to this? The essence of research is to provide solution. Research that is demand driven and also that is need driven, need based, is very encouraging. So, in order to solve that problem in my community, particularly, I decided to utilize this substrate. They also have cow dung, poultry droppings, cassava peels, pineapple waste, and others that can be used as substrate. That was what I used during my primary substrate. This substrate of our waste are nuisance to the environment, and so in attempt, to provide solution to this nuisance, that is what we call to utilize this waste. So, any type of waste can be generated into biogas. Cattle waste and others also have biogas composition. And also, but the maintained value is more important when selecting the substrate. You go for the substrate that has high maintained value and biogas value. In the course of this research, we try to design an anaerobic digester that can, fast track the production of biogas. And there are different types of anaerobic digester. Some of them are provided there, fixed dome biogas plants, septic tanks, or toilets, which behind your door. But most importantly, what we did is based on this. This is our locally designed anaerobic digester. This anaerobic digester, what we're looking for opportunity to measure to determine the quantum of biogas produced. We couldn't find. And then our anaerobic digester was designed, and we, through contact with one of my colleagues, the Deputy Vice Chancellor, Research Education and Collaboration of Peter Okafor, we suggested that we use gasometric chamber, which employs volumetric analysis to measure the quantum of biogas produced. And so, as the gas is produced, it passes through the tube and then displaces a paraffin wax, which is liquid, and then the volume of liquid displaced is equivalent to the biogas generated. And that is how we're able to measure and have our data in the course of our research. This research was supervised by my mentor, Professor James Ipoge, who was a sitting vice chancellor at that time, but he had time to come to the lab to inspect that research and give it speedy, completion. This is another prototype of it. This is our digester in the laboratory in the course of the research in microbiology department, our lab, biogas research lab. This is also the researcher inspecting the volume of biogas produced in the data. This is an upscale of our biogas technology. You have laboratory scale and then panel scale. And so here we're trying to upscale, the technology. The blue drum seated there is about 300 liter capacity of anaerobic digester, which the gas generated there can be trapped by the 500 cubic meter receptacle, which can be converted or can be used and applied anywhere in the world. If you come to Directorate of Science, Engineering and Technology Project, you will benefit from this knowledge. Of course, you are going to pay consultancy fee. Here is simply showing the flaming gas flame of biogas from the anaerobic gasometric chamber, and also this blue flame here is coming from a constant corner in the laboratory, which can easily be used, to sterilize wire loop and other materials in the laboratory. What are my findings? Where are my findings and research on this? One of them is simply that while the number of microorganisms were isolated and sprayed, including fungi, and there was a succession, especially those that cannot survive the anaerobic environment, faded away at the preliminary stages, and then the methanogens survived to break down the substrate, the acetic acid to biogas. These are the methanogens that are able, to break down the methanogen to the organic substrate to biogas. Findings from this show that bacterial population decreased after digestion, confirming that anaerobic digestion could be used to reduce bacterial load in waste disposal. In our attempt to find the suitable temperature, we noticed that 88 to 36 degrees Celsius favored biogas production in our research. Also, the pH, range between 6.0 to 7.8. We try to also determine the effect of starter culture on biogas production. We noticed that digesters that were inoculated, biostimulated with microorganisms for which we derive from cow dung, generated more biogas than without inoculum or starter culture. In trying to determine, production of biogas, we found out that a combination of mixed substrate generated more biogas than individual substrate, so to speak. And then we also determine the weight. Will 1 kg generate more biogas than 2 kg or 3 kg? And we found out that 3 kg generated more biogas, which is an advantage. Thank you very much. Please let me go. So more substrate produce more biogas. That is the result we have here. We also determine the duration of biogas. We notice that biogas production can commence from 5 days with optimum at between 15 to 20 days and then decreases over time. But as it's stimulated with substrate, the generation increases. Here we try to determine biogas production with different substrate. We notice that cow dung generated more biogas than other substrate. Hence we use this as a starter culture. Here is determine the percentage production correlation between inoculum and non-inoculum also was positive. Here, as we told you, cassava contains cyanic acid. This place we have 2 peaks and then it dropped. When microorganisms, cyanic acid was, acted against the microorganism, there was a drop in biogas production. But as the microorganism was able to overpower the cyanic acid, there was a boost in biogas production. And then we also tried to research, apart from our primary substrate, we also tried to find out if other substrates like vegetable, occidentalis, banana peels to produce. And we discovered that this waste can also generate biogas. We also tried African white cocoyam, Protesia osculentum, and mix it with cassava liquor. We also noticed that it could also produce biogas. Also attempted watermelon, mango, and pawpaw, not the real watermelon, the waste that fruit vendors, throw away. We go and scavenge them and use them for biogas production. So, which means no waste is wasted. How can biogas be applied? Biogas produced in this research can be used to power generator. A generator fire the Summit SPG 5000E generator has been powered by the biogas produced in the course of this research. And the implication is that this biogas, produces less carbon dioxide as against fossil fuel, which is more environment friendly and eco-friendly. Here, we can also use the biogas to power a cooking stove, and that is the researcher demonstrating it. And please, if you need, please consult me. The next is that you can use biogas to power a Bunsen burner in the laboratory. Sitting behind microbiology lab is the giant tank for biogas. But since I came in, no gas have been, introduced in there. So which means there is a lacuna. So this research can also, fill in that gap. See, a Bunsen burner. Here is a gas cylinder that has been modified. The substrate is inside, so the gas is generated inside. You don't need to refill it. Just fill in your substrate on daily basis, recharge it, and you have your gas produced, and you can use it on daily basis. Besides using them to generate electricity, we also noticed that the storage was also very, very essential. And so using maize as a case study, we try to see comparing control with the substrate poultry droppings. We found out that they gave better yield, than when the storage was not there. One of my students, in the person of Joyce Akan, use the technology to improve the production of pepper, Asikong frutescens. Rejoice, if you are here, please stand up for recognition. Yes, I co-supervisor in the PhD from soil science. Use it to produce, yes, Viagra seems to be lacking in Nigeria. Maybe because we think that the Viagra is in abundance, but in advanced countries like China, United States of America, Germany, Brazil, they have used Viagras to power their generators. In vehicles are being used, Viagras to be used to power vehicles, and it is possible in Nigeria. Besides Viagras research, I've also delved into other areas. One of them is in isolation. My MSc isolated 4 fungi which have potential to produce extracellular lipases, which was supervised by my mother academic and the person of Reverend Professor Rose Vanna. Please, mommy, can you accomplish with due sense of humility, stand for recognition? She's here to honor me. She supervised. The importance of this is that the fungi associated here have been deposited at the International Microbiological Institute in the United Kingdom, and so they could be extracted from there and utilized. So they are sitting there as sources of knowledge. Also, we try to evaluate the effect of petrol stations in proximity, and we discovered that petrol stations in proximity are sources of pollution because hydrocarbonoplastic microorganisms and fungi were isolated from the environment. Furthermore, we try to analyze or evaluate, the public health importance of dirty loads, and we discovered that dirty loads which we obtained from proximity and Imo State, were potential, disease carriers, harboring good capacity and negative microorganisms. Those of you who use their saliva to count money, you touch the saliva, touch the mouth, count the money, beware, because you are liable to encourage. If you see microorganisms coming directly to you, won't you run away? But you contact them, through practices that you don't know. So this is a way, even bank staff, while they count that money is rolling, just aerosols are generating they generate microfish. So we advise you to use face masks, especially those working in the bank, and also use hand gloves when you are counting money to avoid the problem of microorganism, and you go ahead and make decisions that this is unprofitable. Also, we try to analyze, when I was doing my sabbatical in Latvia, where our mommy was a biosensor, we try to evaluate, certain water, those registered with NAFDAC and those not registered. And we discovered that the 100 of them registered with NAFDAC met international standard, but those unregistered did not meet international standards. So we need to monitor, water quality, not only in Latvia but also in Calabar and Nigeria as a whole, to ascertain their quality before drinking them. So beware of the type of certain water you take. In the course of my research, in collaboration with one of my co-researchers, Professor Michael Etta, we try to analyze the surface area of breast, the nipples of breast area, with respect to the breast milk and how it relates to children. We noticed that the surface area has, various levels of microorganisms which are pathogenic and also will infect the child. So we disadvise that breastfeeding mothers try and practice hygiene, personal hygiene. Wash your breast nipple very well. Some women wear brassiere, remove it, keep it aside, 1 week, 2 weeks without washing it. They still use breast from that brassiere to suckle babies. How will the babies survive? That is why babies from 0 to 5 years don't survive, because of unhealthy practices of their mother. What is my future research focus? My future research focus is to harness the fast waste in Cross River State and in Nigeria to establish a modular refinery Cross River State. This can be established at various local government areas where we have the waste resources. This is a typical example of a model refinery which can be established at various local government areas. And this is the full-scale refinery of biogas, which can be found in other countries. All right. And then, furthermore, our future research emphasis is to make sure to see how we can extract metallurgies, screen them, purify them, and put them on the shelf. So that you don't need to go to the raw cow dung, you need to just pick it from the cell, just like a baking yeast, so that you can inoculate it. But, so that is our future research. And I have a research team called Gas Grow Team. Gas Grow Team, please, can you stand for recognition? This is a team we went to Abuja, Tesco research, and we had a team, and we are planning to produce this metallurgy. Very soon, we are going to play a demo on that. The roles of microorganisms, in the present economic realities. Okay. Here we are going to look at the roles of microorganisms in the present economic realities. Now that there is hunger in the land, pollution everywhere, energy crisis everywhere, what do? We need to tap into the potentials of microorganisms so that we can unravel their relevance. For they have found expression in several areas in global warming, bioremediation, and control of recalcitrant and plastic. Potential of microorganisms can be widely typed as metallurgies. Summary: from the foregoing, inaugural lecturer has been able to clear our doubt, or believe an idea of misconception or fallacy that microorganisms are unprofitable. We have demonstrated and unveiled their relevance in various years in environment to humanity, and also providing solutions to the renewable energy crisis. That will fail me, but permit me to share my research contribution to knowledge. Having found microorganisms, as having during my BSc, MSc, and PhD levels, and publications, several information have been put to the public domain, which many have benefited from. I've also told several students who are contributing to development in the society. I'm interested to know that my research on biogas is the first in the University of Calabar. The pioneer research on biogas is the University of Calabar. It can therefore serve as a reservoir of knowledge and information and references on biogas research in the university, Nigeria, and the world alike. Many researchers have sent their students from University of Ibadan to University of Benin, and even some students from environmental science have benefited from this technology and graduated. The 4 extracellular lipase producing fungi are now sitting in an international microbiological institute. Thank God we now have a cultural center in microbiology department. Maybe I will recall those microorganisms and stop them in microbiology department. Our HUD, our father in microbiology, attracted that status, and our HUD, the sitting HUD, Dr. Morris Etta, is the custodian of that stock culture. Furthermore, we have also used our research on biotechnology to design an anaerobic digester with calcium chamber that can be used in our research laboratory, at various ways. Several recommendations have been made, and what is that is to make deliberate effort to tap into the potentials of microorganisms to maximize their benefits. All right, there should be increased funding of research on biogas technology. Application of biogas methane production in the Bible. There are several applications of microorganisms in the Bible. For instance, when God wanted to attract Moses' attention, he sent metallurgists to ignite the bush, and as the bush was burning, Moses' attention was attracted, and God sent him on an errand. Secondly, when Elijah encountered the beasts of Baal, about 450 of them, the lamb of sacrifice was brought. There were pieces the process of processing, and Elijah told them, add water the first time, add water the second time, add water the third time, signifying the ratio of 1:3 of sulfate remain to water. Then Elijah told them, call your God. But since their God is the dead God, he did not recognize, he couldn't even know that metallurgians are available. Elijah called on the God of fire from heaven, and God released metallurgians, which acted on the altar, the waste on the altar, the pieces of animal, organic waste on the altar, and transformed them into methane gas, which ignited the altar, and the altar busted into flame. So, what do we conclude? God himself is a microbiologist. Conclusion. Pastor Thomas, the Inaugural, has been able to correct the wrong perception that microorganisms are inconsequential to us. Though they have been implicated as causal agents of certain diseases, they can be controlled by simple practice of regular and effective hand washing. It has unveiled their importance in agriculture, medicine, in the environment, and also in humanity. Time will pay me to Pastor Solomon, the special medical, we give it the above for going. I have confidence that I have been able to demystify the myth that microorganisms are wrong for people, and that I have been able to unravel their relevance to humanity, to the environment, and renewable energy solution to solve our energy crisis in Nigeria. So much appreciation to all who, in one way or the other, have contributed to my success and achievements in life. Firstly, I wish to copiously thank the Almighty God for the abundant grace which has been sufficient for me as a destiny child, as the citation reader has alluded, and the most especially for making today a reality, to bring me all the glory. I sincerely appreciate my late parents, Priest Etta Asikong and Mrs. Asikong, who ensured I was nurtured in the fear of God and sacrificed their all to give me a positive division to the university level. They truly laid the foundation for today's inaugural lecture before the demons. I wish to especially thank my darling wife and treasure. It's like though the MC wanted to know the registrar wanted to know she's my treasure. The whole thing I have in the world is deposited in her. Yet, my position in the state, well elaborated by the MC, that will pay me, but more importantly, she is an associate member of the Nigerian Institute of Public Relations, NIPPR. Let me also thank my wonderful children who are beautifully and wonderfully made in Manila, who is Panaye Medical School of Nigeria, Favor, a B.Pharm student in Faculty of Pharmacy, Divine, a student in Law, all in the University of Calabar. Thank you very much, for your support. Let me especially thank my supervisor, Professor James Ipoke, the 9th Vice Chancellor of this great university, who, despite his task schedule, as a sitting Vice Chancellor, found time to supervise my PhD. He sacrificed his time to visit the research laboratory, as you saw in the picture room, offered invaluable advice and instructions which facilitated the work to completion. Most interestingly, Professor Ipoke also taught me biology in the secondary school, Agro Comprehensive Secondary School, which laid the foundation that made me a scientist, a microbiologist. Coincidentally, he is also a microbiologist. So rightly, he qualified to be called a mentor par excellence. Thank you, my academic mentor. I appreciate you for the good job you've done. I also wish to especially acknowledge my MSc supervisor, Professor Rose Tebana, who is physically here to grace this occasion. Let me thank you for honoring this, as a mentor to me. Thank you very much. Utmost appreciation to our predecessor and amiable Vice Chancellor, who found me worthy to be in a management team. whom all who appoint me as the director in the engineering and technology project. I also wish to acknowledge, the former Vice Chancellor. Let me also acknowledge the father of microbiology, Professor S.P. Antai, who is here. All of us are passed through his tutelage. Thank you very much, sir. With the background we did that is making me to stand here, hold the top as a microbiologist. Thank you very much. Time will fail me. I wish to thank my elder brother, Asikong, and all my siblings. I also wish to thank my mommy, Regina Idiban Anari, and my elder auntie, Mrs. Mercy Abu Abam, who is here. Thank you very much. She filled in the gap when they matter. With sorrow, I wish to acknowledge my auntie, Mary Ernest-Bassey, who laid the foundation for my university education, to the late Reverend Father Francis Baba, who is from Akwa State. Also, with sorrow, I wish to acknowledge the contribution of my late uncle, late Mazi Eswalo. Profound appreciation to my cousins, His Royal Highness, Ernest-Bassey Asikong, the fifth, who is the current clan head of Bassey and the Biotechnology Bank of Biotechnology, has introduced all other Royal Highnesses. Thank you. I also wish to acknowledge the presence of His Royal Highness John Hangibo, who is clan head of the party and the acting chairman of the Biotechnology Affairs Council. They are well recognized. I also wish to acknowledge the presence of other Royal Highnesses who are here present. Thank you very much. I wish to thank my in-laws, the DG and East, who have been very supportive to me, including the Boho, the Chinas, Plantain, Godwin, and others. They have been there for me and my family. Thank you very much. My siblings, you are highly appreciated. Time will fail me to mention them. The Tigidi family meeting in Calabar. I welcome the Tigidi family, ESCO. Abi family, are you there? All stakeholders and brothers and sisters, I appreciate all your effort. I wish to thank my supporters who have supported me. In fact, many people from far and near supported and made this inaugural lecture a reality. I've already appreciated Professor S.P. Antai, the father of microbiology. Let me acknowledge and thank the acting dean of Faculty of Biological Sciences. Special thanks to our workaholic head of department, Professor Maurice Etenyong, who worked tirelessly to secure full accreditation in microbiology. Now, my students, microbiology department, members of Faculty of Biological Sciences, thank you very much. Human Resources Department, ICT, Planning Committee, Technical Group, AYE, and members of the Clinical Review team. I want to thank you for this. On this note, thank you. God bless you. Thank you. Thank you. And thank you.
On behalf of the Vice Chancellor of the University of Calabar, Professor Florence Bankubi, and the entire management of our university, I present this memento to Professor Ernest-Bassey Etta Asikong, the 126th inaugural lecturer of the University of Calabar. Congratulations.
It is my honor and privilege to present a copy of my inaugural lecture for keeps in the university treasure.